In this video, we are going to discuss another important process, that is glycolysis, and it is a very important process that takes place during the respiration in plants. Now, we have discussed in the previous video that how cellular respiration helps in breaking down of complex macromolecules, and these macromolecules provide energy in the form of ATP. Now, this breakdown of cellular respiration or the breaking of macromolecules can be into two types, that is aerobic and anaerobic. And in both the types, whether it is aerobic or anaerobic, the first step is the glycolysis, in which a 6 carbon atom molecule is broken down into two molecules of pyruvate or pyruvic acid, and that is a 3 carbon atom compound. Also, it is very important to know that the process of glycolysis, it is common for anaerobic breakdown as well as aerobic breakdown, both the processes, and it takes place in the cytoplasm of the cell. Also, it is important to note that this process does not require oxygen. Here, the glucose undergoes partial oxidation to form the two molecules of pyruvate, a 3 carbon atom molecule. After the formation of the pyruvic acid or the pyruvate, the fate of the pyruvic acid depends upon the presence or absence of oxygen. That is, whether it will enter into the Krebs cycle, that is the complete oxidation resulting it into the formation of carbon dioxide and water, or whether it will undergo the incomplete oxidation or incomplete breakdown, that is fermentation, and the products are completely different, that is ethyl alcohol is formed or lactic acid formation may be there. That we'll study in the later videos. Now first let us talk about glycolysis. This was just an introduction part. Now let us talk about glycolysis. Glycolysis is actually a series of closely related reactions, and in which a hexose, that is normally glucose we are talking of, it breaks down, and the steps of glycolysis are also known as Embeden-Meyerhof-Parnas pathway or EMP pathway, because of the scientists who trace these processes. So this is glycolysis is also known as EMP pathway or Embeden-Meyerhof-Parnas pathway. When we study these steps are subdivided into two major phases. The phase of breakdown of glucose to 1-fructose 1,6 biphosphate is phase 1, and here onwards this is phase 2. So the process of glycolysis or EMP pathway has been broadly divided into two phases, that is phase 1 and phase 2. Now, the process starts with the conversion of sucrose first, which is a 6 carbon atom, and this is the product of photosynthesis. So, the sucrose is first converted into glucose. The entire process of glycolysis happens in 10 steps, and each step is guided by enzymes. So there are 10 steps to the glycolysis process, and there are 10 enzymes which are involved in the process of glycolysis. So the first step involves the conversion of sucrose, that is the product of photosynthesis, into glucose in presence of an enzyme invertase. The next step is the phosphorylation. Here, glucose is phosphorylated and in the presence of ATP and the enzyme hexokinase. The conversion of glucose or phosphorylation of glucose takes place over here. So let us write phosphorylation over here. Phosphorylation takes place, the first phosphorylation takes place over here, and utilizing the ATP and in presence of enzyme hexokinase, glucose 6 phosphate is formed, and along with it ADP is formed. Second step, the glucose 6 phosphate gets converted into fructose 6 phosphate. So this step is isomerization, because here only the position, both are 6 carbon atom structures. We are starting with 6 carbon over there, and glucose 6 phosphate gets converted into fructose 6 phosphate in presence of enzyme phosphohexoisomerase, and this is isomerization. This fructose 6 phosphate undergoes phosphorylation again. So this is isomerization. This is the second phosphorylation. And again here the ATP will be used to utilized, and in presence of enzyme phosphofructokinase, the fructose 6 phosphate gets converted into fructose 1,6 biphosphate. Now this is again an important reaction. Also over here the presence of magnesium ion is required. This reaction takes place in presence of magnesium ion. From here, that is from the sucrose we are talking, gets converted into glucose, then glucose 6 phosphate gets converted into fructose 6 phosphate, and it gets again phosphorylation happens and it gets fructose 1,6 biphosphate. This marks the completion of the first phase of the glycolysis. Now the second step, that is the formation of pyruvic acid from fructose 1,6 biphosphate. From here it involves 6 steps. So this is the first step. Then we talk of second step, third step, fourth step. So this is first, second step, third step, fourth step. Now here we are, this process involves 6, the second phase. So here 5, 6, 8, 9 and 10. So here we have 10 steps in glycolysis. 1, 2, 3, 4, 5, 6, that is 6 steps. Now here the next phase, the fructose 1,6 biphosphate gets divided into or splits into glyceraldehyde 3 phosphate, or it is interchangeable, and dihydroxyphosphate. Now this is the 6 carbon atom has been broken down into 2, 3 carbon atom structures, and these are interchangeable in presence of enzyme aldolase. Here the enzyme phosphotrisoisomerase is also responsible for converting fructose 1,6 biphosphate into dihydroxyphosphate and glyceraldehyde 3 phosphate. Now here the next step is the formation of 1,3 biphosphoglyceric acid. Now here you can see the 6 carbon atom has been broken down into 2, 3 carbon atom, and further it is carried as 3 carbon atom till the end. So the glyceraldehyde 3 phosphate gets converted into 1,3 biphosphoglyceric acid. Now here this reaction involves the addition of inorganic phosphate and the reduction of NAD plus to NADH and H plus. So here the reduction is taking place. So we call it as phosphorylation, and here again phosphorylation and oxidative dehydrogenation. This is the next step. Here, in this step, 1,3-biphosphoglyceric acid is converted to 3-phosphoglyceric acid in presence of enzyme phosphoglycerokinase, and the first molecule of ATP is formed. This is very important because here till now we have been utilizing the ATP, that is energy is being used, but here the ATP is produced. So here the ATP molecule is produced, and the first molecule of ATP is generated when 1,3-biphosphoglyceric acid is converted into 3-phosphoglyceric acid in presence of enzyme phosphoglycerokinase. The production of ATP from ADP in this is known as substrate level phosphorylation, because here there is no using of NADH. We are not utilizing NADH over here for the conversion, and therefore this is substrate level phosphorylation. The next step, that is the conversion of 3-phosphoglyceric acid into 2-phosphoglycerate, happens in the presence of enzyme phosphoglyceromutase, and it is an important step. Again, the 3 carbon atom structure is there. Next step, that is the 2-phosphoglycerate gets converted into 2-phosphoenol pyruvic acid or pyruvate, and here this process is dehydration because a molecule of water is eliminated. So here the molecule of water is removed from this glyceric acid, 2-phosphoglyceric acid, and it gets converted into 2-phosphoenol pyruvic acid or pyruvate. This happens in presence of enzyme enolase. Thus, 2-phosphoenol pyruvate gets finally converted into pyruvic acid. 2 molecules of pyruvic acid are produced. Now, this happens in presence of enzyme pyruvate kinase, and again here, second time the ATP molecule is produced. So here, in the case of glycolysis, when we talk, there is eventually 2 molecules. The pyruvic acid, 1 molecule of glucose form 2 molecules of pyruvic acid, and in the first phase, that is over here, if we just see in the first phase, that is the conversion of glucose to 1,6-biphosphate, that is fructose 1,6-biphosphate. There is no energy gain because here in the first phase we are only utilizing the ATP, not producing the ATP. But in the second phase, that is from here to here, there is production of 4 molecules for each conversion. There is production of 4 molecules of ATP during each phosphorylation. So what is happening actually over here? We are producing, when we are talking over here, the net gain of ATP which is being produced over here, in the first phase, we are using 2 ATP molecules because over here at the first step it is used, and in this step also it is being used. In the second phase, we are producing 4 ATP molecules for each conversion. So the net gain is 2 ATP produced because we are producing 4 and producing or are utilizing 2 ATP. So the net gain is 2. So thus in the conversion of 1 molecule of glucose, 2 molecules of pyruvic acid are formed, The net gain of ATP is 2 ATP because 4 ATP are produced during the second phase, 2 in each type, and 2 are utilized during the first phase. So net result is 2 ATP. Also, there is a net and there is formation of 2 NADH. So here the glycolysis is an important process from the which results into the breakdown of the glucose 6 carbon atom into 2 molecules of 3 carbon atom structure, which is known as pyruvic acid. And then from here onwards, the fate of the pyruvic acid then enters, whether oxygen is there or there is anaerobic breakdown, it then goes finally into the Krebs cycle. If it is aerobic, in case of aerobic breakdown, or it may undergo fermentation in an anaerobic condition, that is absence of oxygen. The glycolysis is an important process and is the beginning of the breakdown of molecule of glucose.